Um, so hello everyone, my name is Shou-Wen. I'm an assistant professor at Westlake University. So today I'm going to share with you a story about lineage tracing. So speaking of lineage tracing, speaking of lineage, we can first think about family lineage and also genetic lineage. Actually, this is one of the early experiments by Manda, where he proved the rules of modern genetics. And however, I'm very, very fascinated about cell lineages. So this is the relationship of individual cells within a living organism. Using the first two lineages actually lays the foundation of the modern biology. And the last example, and this is actually C. elegans lineages, is generated by John Sutton about like 40 years ago. And from this lineage, he actually found the phenomenon of apoptosis, which awarded him the Nobel Prize in 2002. So what we can learn from lineages, the first thing we can learn is how cell differentiates, especially a stem cell. And second thing is we can study how individual cells proliferate. Also, how cells can migrate from one tissue to another tissue. And lastly, the memory of cells. So I think that individual cells, they are all different, and each cell has their own molecular features. And this kind of molecular feature can propagate to its future generations, which I call inheritance or memory. So these are four building blocks, what we can learn from lineage tracing. And in specific biological questions like cell-fate determination or cell-fate conversion or tissue development or regeneration and disease progression and so on. So some or all of these building blocks will be involved in such phenomena. So lineage tracing actually has revealed some very interesting biology. Next question is how we can record cell lineages. So we can simply barcode or label individual cells and trace them all the time, and then we can see like what kind of cells are shared within the same lineage. In 2010, people use different colors to label individual cell lineages in the in testing. And nowadays we can have a much better throughput. We can use different genetic barcodes to label lineages. For example, we can generate some random DNA sequences and using lentivirus to integrate these random sequences into the genome of the cell to label the cell lineages, and this can achieve very, very high throughput. And also, you can simultaneously profile the lineage barcode and the gene expression at the same time in a single cell. And then you can also introduce a barcode array into the cell and use Cas9 to add this array over time to generate lineage information. So one of the earlier interesting piece of biology we get from lineage tracing is we can show that by labeling LGR5 positive cells, they actually give rise to all different downstream fate outcomes in the intestine tissue. And so therefore we conclude that LGR5 positive cells are intestine stem cells. Another interesting study is in the context of hematopoiesis. So we, by using transplantation assays, long time ago, people have established that HSCs can give rise to all the different downstream fate outcomes. However, this is a result established from transplantation. So it's very unclear whether HSCs directly generate all the different downstream fate outcomes in a native context where we do not have any perturbations. So about 10 years ago, my collaborator Fernando Camargo, he actually used a lineage tracing mice to barcode all the different cells, and he found that actually in the native context, long-term HSCs do not actually generate all the downstream fate outcomes. Therefore, reveal a very different picture between in vitro and in vivo systems. The previous two studies, they used relatively preliminary lineage tracing technology. Nowadays, we can use single cell technologies to do lineage tracing. So in the study by my postdoc lab along Klein's group, we can first barcode a cell and then allow the cell to divide to microclines. And then sample one of the cell to get both its transcriptomic state and also lineage information, and then allow the other cells to continue to divide and differentiate to give us the future data outcome. So this is the single cell RNA sequencing study. So this is the transcriptomic embedding for all these about 100,000 cells. You can see here the first one, these are two clones. So actually they barcode cells at day 0 and collect the cells at day 2 and day 4 and day 6. So in the upper clone, so this clone actually progressed almost linearly from the progenitor stage to future fate outcome. And then in the second case, cell it actually progress and give us to different fate choices. So we can actually also use this to study the cell fate choice of the individual cells. Here I'm showing all the day 2 cells and color them by their future fate outcomes. So these blue cells will eventually give us to monocyte, and the red cells will eventually give us to neutrophil. And so suggesting that you can, looking at this map, you can already see that even very early on, cells already make their different fate choices. Another very interesting finding from this study is that they use machine learning models to ask whether the transcriptomic stage of the day 2 cells can already predict their future fate outcome. However, the answer is no. The accuracy is only about 50%, suggesting that there are some underlying biological information not captured by the transcriptomic information that will determine the future fate outcome. So we guess that this kind of underlying fate determining factors could be some epigenetic information. So indeed, a single the fate choice of cells is very complex. It can determine depend on an external factor and an internal factor. And for the internal factor, not only the gene expression, but also the state of the protein and the protein expression and so on. Indeed, in a study from 2014, you can see that they profiled the hematopoietic systems and sort different populations to do RNA sequencing. And then they classed the different populations by their RNA expression. And you can see that this MEP is classed together with the HSC, which is the hematopoietic stem cells. However, this MEP is going to generate the erythroid cells. Therefore, using the RNA embedding, the MEP, the proteins of the erythroid cells, is actually more closer to the stem cells. But very interestingly, if you use the H3K4 methylation 1, a marker for the histone modification, now the MEP is very close to the erythroid lineages according to the H3K4 methylation 1 information. And this suggests that using the epigenomic information, sometimes it can foreshadow the future expression of the genes. And this may help the prediction of the cell fate choice. So, however, currently there is not yet any technology that is able to simultaneously profile lineage barcodes, transcriptome, and epigenome in single cells. And this kind of technology is actually very needed in order to systematically understand how cells make their fate choice. Another limitation in the current field is the lack of a good model system to study cell lineages in vivo. So this is a few years ago, the Fernando group, they provide such a technology called calling, where they can use the dots to induce every individual cell in the mice to generate some random barcodes. And this kind of random barcodes can be expressed as mRNA, and therefore we can simultaneously capture both the lineage barcodes and the transcriptome in a single cell. Specifically, so there is in the genome, there's a target regime, and also there's a guide RNA. So the guide RNA and also the Cas9 can be expressed upon DOX induction, and then edit the target region to generate both insertions and deletions or their combinations. So these are just heritable random barcodes that can label individual lineages. Unfortunately, there's only about 44,000 different lineage barcodes compared to over 10 to 10 cells in an adult mouse. Another limitation is that so the efficiency to generate random barcodes in this target region is only about 30% when you introduce DOX editing. And also, because these random barcodes has very low expression efficiency, so when you try to measure them using single cell assays, you can only capture only 40% of cells will detect these lineage barcodes. So in combination of this low editing efficiency and low capture efficiency, so only about 10% of profile cells will have edited lineage barcodes. And this means that if you profile 1,000 cells, only about 100 cells will be useful for downstream analysis. So a few days ago, we published a new paper in Cell where we introduced two technologies to solve this problem. The first one is the starling mice, and it has a lot more, which is 10 to 18 theoretical lineage barcodes, far more than the total number of cells in adult mice. The second advantage is that using the starling mice, now if you profile 1,000 cells, 700 or 70% of the profile cells will have valuable or useful lineage barcodes. Another technology is the community where we can simultaneously measure both the lineage barcodes, gene expression, EMI station, and chromatin accessibility in a single cell. So first, I will introduce the starling mice. We actually, in order to improve the first generation starling mice, so we look at the data and we realize that there's a lot of large scale deletion in the starling mice. So for example, the total length of the target array is 270 base pairs, but most of the deletions is about 200 base pairs, which means that about 2/3 of the whole entire sequence has been deleted. And this is compared to just about 0 or 1 base pair insertions in this target region. And with reason that we can introduce a lot more insertions, and this could greatly improve the barcode diversity. To do this, we fuse the Cas9 and TDT protein together. And TDT is a protein, an enzyme that is used in the immune cells to generate diverse BCR receptors. So indeed, by fusing these two proteins together, we observe that now we can achieve about at least 1 million different barcodes in the collagen 1-1 locus. And furthermore, we expand the original target array from the collagen 1-1 locus to both the tiger and the ROSA 26 loci across different chromosomes. Now we have 3 copies of independent target arrays across different genomic loci, and each one we hypothesize that it can generate at least 1 million different barcodes, and therefore by combination, so we expect to see about 10 to 18 different lineage barcodes in these starlings. So finally, we also are very concerned about the lineage readouts. Suppose we collect a lot of cells. First, we need to, so some of cells we will not be able to detect the lineage barcode at all. So we need to remove these cells first. And then secondly, some of cells, even we see lineage barcodes, the lineage barcodes is just unedited original target array sequences. They will not be useful to identify cell lineages. And lastly, even if the target array has been edited, however, some of the sequence are just some of the mutations occur very frequently. Therefore, it's also not useful to label images. So we also need to exclude these common image barcodes. So, Carling actually performed a relatively mediocre across all these 3 steps, eventually leading to just 10% of the cells that will be useful for downstream analysis. And this is compared to about Darling on the other hand, achieved very good score in each stat. So eventually, 60% of the cells will be useful for calling downstream lineage analysis. So the second technology is chameleon-seq. To enable single-cell multi-omic tracing, so we profiled both the lineage barcodes and gene expression from the cytoplasmic mRNA. And then we also separate the nucleus and generate the DNA methylation formula sensitivity measurement from the nucleus. We applied our technology to 3 different biological problems. The first one is cell-based choice or differentiation. We studied the differentiation in the context of hematopoiesis. Here we introduced the lineage barcoding at E17 with dogs, and then waited for about 2 months and profile the cells from skull. And this is the skull blood cells, and the gray area is the early progenitors, and it can generate all different fate outcomes. These are 3 representative clones. The first one is a multipotent clone, meaning that this clone occupies both the stem cell region and also all different mature phase. The second clone specifically reacts to megakaryocyte. And the last clone is monocyte bias. We have about 1,000 such clones. And most of the clones is very small. Indeed, actually, about 2/3 of them have only 1 cell. So in order to handle this very sparse data, we need more sophisticated tools. So I turn to my the tool that I developed in my postdoc, which is CoSpark. So here, if you give me the data with both lineage measurement and transcriptomic information, so I can use CoSpark to infer the transition rate from a given initial state I to a later state J after a certain period of time. To do this, I imposed 2 assumptions. The first one is sparsity, meaning that I assume that as the cells differentiate from a progenitor state to future fate outcomes, it will progressively restrict its fate choices. And then the second one is coherence, meaning that if 2 cells are very similar in their early stage, they will also have very similar fate outcomes at a later time point. So combining the clonal data, which has like 1,000 different clones, and also these 2 assumptions, so I was able to, CoSpark is able to identify the transition probability matrix and use that to identify the trajectory that a cell will transfers to eventually give to the given cell type. And you can also study how gene expression will change along this trajectory. Using CoSpark to study this data, so first we can see that we can quantify the lineage coupling between different cell types. For example, the HCC and the megakaryocyte have very strong coupling in this data, and also between monocyte and LMPP. So then I can also apply CoSpark directly to predict for each individual cell what is its probability to give rise to, say, megakaryocyte or monocyte. And you can see that for megakaryocyte, so this part of the HCCs have much higher probability to generate megakaryocyte. So we turn to the raw data to test whether this is indeed the case. So we select the clone that only have the observed fate bias to give rise to megakaryocyte, and we ask where the early progenitors come from. And indeed, these early progenitors of these clones are localized in the lower part of the HCC region. And also similarly for monocyte, you see there are a lot of clonally related cells for monocytes that are enriched in this LMPT region. Okay, so the second application is this migration. So we also use the hematopoiesis system to study the cell migration in the body. One thing we are interested is how so one thing we are interested is how the onboarding stem cells, they can potentially migrate from one bone to another bone. So earlier, people have studied the migration of the HCs using a parabiosis mice, meaning that you can connect the bloodstream of two mice together so that the blood cells from one mice can flow to the other mice. However, this is a very invasive surgery and can induce inflammatory response in these mice. Now we have done, we can just induce the barcoding, and now we can study the migration of cells in a very in a normal unperturbed condition. So to do this, we barcode the cells at 8 weeks old, and then wait for about 4 months and collect cells from different bones, the skull, spine, left leg, and right leg, and then use bags to sort HCs, MPP, myeloid progenitor, and microglial, and so on. And then using the bulk protocol, we then profile the target lineage barcodes in this population, these bulk assays. We can see that so in this data, the first letter is the bone origin, the second letter is the different populations. You can see that this is again the lineage coupling between different bones. You can see that different bones are very well. The lineage coupling between different bones are very well separated. What does it mean? It means that the blood production in different bones are have very similar lineages, and within the same bone, the blood cells share similar lineages, but for different bones, the lineage is very different. This means that the blood cells is produced locally. We can also measure or quantify the fraction of the clones that are shared in one bone and other bones. So, for example, for this myeloid progenitor, so we can ask for the myeloid progenitor clones coming from skull, what fraction of these clones are also shared with myeloid progenitor from other bones? And this fraction is quite high; it's about 40%. But if we look at HCs, there's only about 5% of HCs in one bone that are shared with other bones. And this is actually consistent with our understanding that hematopoietic stem cells are, especially long-term hematopoietic stem cells, they are more quiescent. They tend to stay in one bone. But still, we see about 5% of migration activity across different bones if we wait for 4 months. Finally, so I'm very, very fascinated about our application in this memory context. What does it mean? So we can use the gene expression as an example. So we know that cell can express a gene stochastically. Some genes, a gene can be turned on and turned off over time. If this happens too fast, the time scale is smaller than cell division, and then the expression of a gene cannot be propagated to its future progenitors. But if a gene can be turned on and maintained for a while, so that the cells already divide, and then this gene we can call it heritable gene, because a stochastic turning on event can last for a while to impact its progenitors. Indeed, you can some of these genes are interesting. For example, it can confer drug resistance ability for these cells. So instead of studying the memory, so basically this is like a quantifying. You can quantify the memory strengths of individual genes. And some genes have stronger memory, some genes have less memory. But you can also systematically study the memory capacity of the entire transcriptome. So this is again my postdoc lab study, and here they barcode cells on day 0, and by day 2, the cells from the same clone have already divided 3 times. And now each color is a different clone, and you can see very visually that each clone has distinct color. Each clone have very similar transcriptomic states. And this is the evidence of memory, like the cells coming from the same founder cell, they have much similar transcriptomic memory than 2 different clones. Another interesting observation from this study is that if you look at the monocyte, and the monocyte can actually be divided into 2 different subpopulations. So the upper branch express macrogenes related to dendritic cells, and lower branch express macrogenes related to neutrophil, but they all express macrogenes for monocytes. So that's why they are called monocyte cells. And if you look at where these blue cells, upper branch coming from, they actually come from these blue cells on day 2. And then the bottom branch coming from these red cells on day 2. And you can see even by day 2, there's a strong separation between these blue and the red cells, suggesting that they are already suggesting that as the cells on day 2, they already have some kind of like differences, and then as they differentiate to give rise both monocytes, they carry the memory of the origin with them even after differentiation. So our question is, first, how long does this transcriptomic memory or transcriptomic clonal memory can last? The second question is, what about epigenetic memory? So far as I know, there's not a single study, lineage tracing, a single cell lineage tracing study to evaluate the epigenetic memory. So in order to answer this question, we can also turn to the homophobic system, particularly focused on stem cells. So homophobic stem cells, they arise from about E10 in the embryonic development through endothelial to hematopoietic transitions. And then once they have born, they can migrate to the fetal liver and undergo massive expansions, and then to the different bones in about when they migrate to different bones later. So we designed an experiment to barcode the HCs at E10 when they just begin to form, and then wait for 36 hours. At this point, they are still in the AGM region, and then or wait for 5.5 days when they arrive to the fetal liver, or wait for about 2 months when they are already in different bones. And for each case, we can profile HCs with chameleon seq. And in this case, we get the lineage barcode, gene expression, chromatin accessibility, and DNA station for each individual cell. Now we can ask the question of memory. So for example, if we take the transcriptome as an example, and we can look at the similarity of 2 different cells in terms of the transcriptome in high dimension, and ask how different 2 clones can be. So in the first case, the 2 different clones have very they don't have clear pattern. So the cells from different clones are mixed together. So this is the case with weak clonal memory. And if a clone has a very strong memory, it means that all the progenies from the same clone, they have similar molecular signatures. Because of these shared molecular signatures, they are very more similar in high dimensional space than cells from different clones. So look at the transcriptome, you can see that, so x-axis is the time, so starting from 36 hours to 5.5 days in the fetal liver, or 10 weeks in the bone marrow. You can see that as time passes by, the memory of the transcriptome degrades over time. However, on the other hand, the protein accessibility, it has almost no memory, or it appears just like a random noise. And interesting for DMI station, you can see that by 36 hours, there is already pretty strong memory in DMI station. And then even in the 5.5 days in the fetal liver, you can see that the memory is very strong. And surprisingly, in 10 weeks or about 2 months after, like over these 2 months, the HSCs, they need to migrate from fetal liver, from AGM to fetal liver and to bone marrow. So, and also each individual clone expand massively, and all the cells from the same clone, they potentially undergo different external environment and stimulus. But still, we can see that the DMI station memory is very, very strong, which is quite surprising. So we conclude that the long-term clonal memory is maintained in DMI station, but not in gene expression or cloning accessibility. So now we can zoom in this particular time point and to see what is going on. So for gene expression, you can see that, by the way, each color is a different clone. You can see that for gene expression, all these clones are mixed together, and also for cloning accessibility. But for DMI station, so visually, you can see that some clones are some cells, for example, this clone, all the cells from the same clone, they are very, very similar. And also for this green and blue clones. Also we did a statistical test to ask whether cells from the same clone have much higher similarity than cells from than just two random cells without clonal lineage, clonal similarity. And indeed, you can see that in DMI station we have significantly strong similarity within the same clone, but not for cloning accessibility or gene expression. In total, we identified about 300 genomic regions that have differential DMI station. And these are 3 examples. And you can see, for example, here, clone 149 has about, in this particular region, has about only 10% methylated. And then the green clones have about like 20% methylated. So our remaining question is, first, what is the biological implication of this clonal memory? And second, whether this phenomenon can be observed in other cell types. Remember that our study is only for HCs. The last question is how long this epigenetic memory can last. So currently, by 2 months, we can still see a very strong memory. But how about 1 year? And yeah, to summarize, we developed a Darling mice is a high-capacity tool for in vivo lineage tracing in mice and across different tissues. And we also developed Epigenetic, the first single-cell multi-omic lineage tracing method to profile both the gene expression and the epigenome and lineage together in single cells. And lastly, we provide the first direct demonstration of strong epigenomic memory at single-cell level with lineage tracing. And so this work is done by Dr. Lily at Boston Children's Hospital, and she's the first author. And this is my collaborator, Fernando Camargo. And thank you very much.